Sorry. So I'm Annalisa Calamida, and I work at the Space Telescope Science Institute. I'm the Deputy Branch Manager of the Wi-Fi Camera 3 instrument on HST team. So in particular, I focus on improving the flux calibration of both cameras. We have a UV optical camera and an infrared camera. And I also collaborate with other scientists, Space Telescope, work on other instruments, and JWST and Raman to homogenize the flux calibration of the instrument. So if you are interested in calibration, you can come and talk to me. Today, I will present a work based mostly on ground-based data collected with a Wi-Fi camera, which is DECAM, on the Blanco 4-meter telescope, and how we can, it's an example science case on how we can use wide-field ground-based photometry coming from these telescopes and surveys and combine to very accurate and deep photometry from HST and now also JWST to study galactic globular cluster and in the future possibly other local group cluster or even extragalactic globular clusters. Why do we care about globular cluster? Well, they are present in most galaxies, as we've seen in previous talks as well, and they contain the oldest stars in the universe. And so, for example, giving an age estimate of globular cluster gives also a lower age estimate for the universe that can be compared with other estimates. And in stars in globular cluster are all at the same distance and more or less they have the same reddening, and they also have the same age and possibly the same metallicity. There are a few exceptions, as you will see. And stars in globular cluster recently from spectroscopic studies, mostly performed with VLT, we saw that all the stars in globular cluster show a light element abundance spread and some anti-correlation of the light elements, for example, oxygen and sodium. And there are still many open questions about globular clusters. When I started my PhD, we thought that these were simple stellar population, but actually they are not. So there are multiple generations of stars called subpopulation with different abundances, and we don't know why every cluster is different from one another. And then we still have to understand the complete picture on how the globular cluster system of the Milky Way form, and this could shed light also on how other globular cluster systems form in other galaxies. So, okay, I put this slide, but I guess most of you or all of you already know what a color magnitude diagram is. This is the tool that I select today to study this globular cluster. It's a very powerful tool. So you see stars in different sequences. Most of the stars will be on the main sequence where they burn hydrogen in the core, obviously. And then we have the red giant branch is the following sequence. The horizontal branch, because in some filters, not all of them, this phase is actually the stars are horizontal and is used for calculate the distance of the star. For example, in the bulge, yesterday we saw the red clump stars are used for distance as distance indicators. And then we have many and thousands of white dwarfing of the cluster, which are so faint that it's very difficult to detect. But in some cluster we call with HST list. So the first cluster I will present results on is 2808. I said this is the second most massive cluster in our galaxy, and it doesn't have any iron spread like a typical globular cluster, but HST photometry showed that the main sequence of this cluster was split in 3 different sequences, thought to be different because of their helium abundance. It shows the typical light element enhancement and oxygen-sodium anti-correlation of the clusters, globular clusters. So this is my data set. As you can see, DECAM has a 3 by 2 degree field of view, so it's huge, and it uses the typical Sloan-like filters. In this case, we have exposure in U, G, R, and I band. And then I combine this data set with HST data for the cluster core, because essentially for DECAM, this is a too crowded region, and we couldn't get accurate photometry in the center unless we use HST. And the aim of this data was to study the spatial distribution of the different subpopulation in this cluster, and also eventually determining the density profile of the cluster and see and verify for the presence of extra-tidal stars. So this is the color magnitude diagram based on DECAM photometry for different colors. And as you can see, we have an issue here because as soon as we go wide with our observation of a cluster, we have also a lot of thick contamination. So you can see the blue horizontal branch stars here, but you don't see anything else from the cluster really. We have Gaia proper motion and they work really well, but they work down to just above the turnoff. So we tried to use Gaia, but then we were losing essentially all the blue horizontal branch stars, and we were very interested in that evolutionary phases. So what we did is to use the power of the U-band filter because it gives us a better temperature sensitivity and metallicity sensitivity. And so we can separate feed and cluster stars, since mostly feed stars are more metal rich than the globular cluster stars in this case. And the method of using color magnitude planes is very well described in these two papers, Calamida et al. 2017 and 2020. I didn't have enough time to go into all the details, but as you can see, it works really well. This is the clean color magnitude diagram, and finally, you can see the cluster sequences. In this case, for this cluster, the horizontal branch is very interesting because it has a populated red horizontal branch, almost no outlier stars, and there's a very extended blue horizontal branch, which can be divided in three groups, as I will show you later. So we defined this index, I mean, it was defined already in the literature, that we call SUGI or CUGI in Italian. And we defined the same index with HST magnitude. So we divided our data in central region of the cluster, so for a radius less than 1.5 arc minutes, and all the rest of the cluster. And the cluster radial radius is about 15 arc minutes. And as you can see, this index is a little bit different, as you can see from the color magnitude diagram here, because in the case of HST, the red horizontal branch goes on the other side, while it doesn't in the DECAM colors. But anyway, it has the same effect. The stellar subpopulation with different sodium and oxygen abundance, they split. And by eye, you'll see, oh, now instead of one red giant branch that I was seeing here, I see two or more. So to really understand how many sequences we can identify in this cluster, we rectified the red giant branch in this color, and we calculated the delta color, and we use a Gaussian mixture model fitting, and we identify in both samples, HST and then DECAM, three main subpopulation. We called the bluest one P1, or primordial population of stars, and then we have an intermediate P2 population and enhanced population. But enhanced what? So we had a lot of spectroscopic data, mostly from high resolution from, I think it was Flames and Giraffe from VLT, from Carretta, and we also had data from Muse published in Latour. And so we could compare this selection based on photometry to spectroscopy. We can see that the primordial stars here in blue are the ones that are sodium poor and oxygen rich, and then they, with time, they evolve and they become rich stars, or P3, these red ones, which are sodium rich and oxygen poor. So we see that this method works, and you can essentially identify all the groups, including all the stars that you can see in the cluster, while with spectroscopy you only have a subsample of stars. So this is a subsample that we could cross-match with the spectroscopy. And then we were able to study the spatial distribution of all the population. As you can see from the histogram up here, the enhanced population is more centrally concentrated, as is expected usually in globular cluster and was observed in other clusters. But then it has a more extended distribution because all the other studies were mostly based only on HST or VLT data, so they were focused on the center of the cluster, and they didn't have the big picture for the entire extent of the cluster, which is up to 15 arc minutes, like you see in this plot. And from the density plot here on the right, you can see that clearly that the density distribution of the enhanced population is more extended, and also the center of this group is shifted in comparison to the primordial population by about 0.1 arc-minutes. We also wanted to study the distribution of the stars in different evolutionary phases and give us information on how the cluster formed and evolved. So in this case, we divided the horizontal branch in the canonical three groups defined in literature, and they're called ABT1 for the blue stars here, ABT2 for the green stars, and ABT3 for the yellow stars in the sample from HSC inside of the cluster and DECAM outside. And what we see is that the red horizontal branch stars, here in red, distribution, so the number of these stars increases going from inside the cluster to outside, as you can see. And while the number of the blue stars here decreases while going from inside the cluster to outside the cluster. The total number of horizontal branch stars along the cluster from the center to the Tadia radius stays constant and confirm previous result based on wide-field camera data and HST data as well. So we said, is this something only peculiar for 2808, or other clusters show the same behavior? And that's true. Other clusters showed a similar behavior, in particular Omega Sun, which is the most massive globular cluster that we have in the galaxy. I say globular because might be that it's not a real globular cluster. It shows more than 1 dex of iron abundance spread, which is a lot, even similar of the abundance spread of a dwarf galaxy. So now it has a retrograde orbit, so it's believed that it's connected to the Gaia Enceladus galaxy that merged with the Milky Way billions of years ago. And or that is the result of the merging of some cluster and in these dwarf galaxies, and then everything was accreted by the Milky Way. And as you can see, the color magnitude diagram of this cluster is a bit different from 2808, since the red giant branch already splits in sequences in a normal color magnitude diagram. We don't need any index to actually see the split of the red giant branch. This is mostly due to iron abundance differences of these red giant branch stars, and it was confirmed also by spectroscopy. I had the spectroscopy abundance histogram here, but it's covered by, I didn't realize the PDF would cancel the plot. But. So this is the DECAM color magnitude diagram for Omega Sun. It's already clean from the contamination of fish stars. We use the same method, color magnitude planes. And this was, when I saw this color magnitude diagram, I was super happy because these are data from a 4-meter telescope. And for the first time, we managed to actually see the split of the mean sequence with this telescope. This was identified in the 90s with HST ACS data for the first time, and was confirmed with VLT data, which is an 8-meter telescope. And finally, we will be able to see it with a 4-meter telescope in very good observing condition, obviously, but this demonstrates the power of these telescopes. And in the case of Omega Sun, we just use the color distribution here of the red giant branch to separate in the most metal poor stars, metal intermediate. We identified 2 groups and metal rich stars. And then we studied the density distribution of this group. As you see from the density plot, even in this case, the most metal-rich stars are more centrally concentrated, but then they show a more extended spatial distribution. And in this case, the center of the metal-rich and the metal-intermediate population are shifted by more than 1 arc-minutes. This is Omega Sun is a much more massive globular cluster, and the Tadia radius is 1 degree in comparison to 15 arc-minutes for 2808. And regarding the horizontal branch stars, we see something similar. Omega Sun doesn't have a red horizontal branch, as you can see, no red horizontal branch stars. We have tons of Arlary stars. And what we see, though, is that in this case, the number of horizontal branch stars stays constant all over the cluster, but the number of ABT1 stars increases, And in this case, the number of ABT3 stars decreases like in the case of 2808. So we see a similar behavior. And here we were able also to trace the spatial distribution of the main sequence stars because this cluster is closer. So we were able to identify the main sequence and the splitting along the main sequences. You can see HSE and VLT told us, yes, blue main sequence stars are more centrally concentrated. They are possibly helium-enhanced, but then they didn't have the full picture up to 1 degree and more, which shows that actually the frequency of these stars is increasing towards the outskirts of the cluster. So concluding, we performed this study only on 2 globular clusters. We see a similar behavior in which the most enhanced stars are more centrally concentrated, but then they have a more extended spatial distribution. So now we need to confirm these results. Possibly we are looking forward for wide field spectroscopy service, in particular the result from the foremost galactic community service and the Moon's Milky Way survey, because we need spectra for the stars in a big area, especially omega Cen is a tidal rise of 1 degree. So we really need that. And also this is only the beginning, because we have many more data available for globular cluster collected with DACAM, but also with MegaCam. And then also I dig the ESO archive and I saw there are data collected with OmegaCam on VST for many other clusters as well. So if you have any student interested in this kind of work, please let me know, because I cannot do everything by myself. We are actually 2 people in the Space Telescope working on this project right now, so we really need help. And then this talk was in particular to show you the power of combining space photometry for the cluster core to wide field data coming from this ground-based telescope and service. And in the future, we will have LSST that is coming up and Raman. And also we will have ELT and JWC that will help us to study the core of globular cluster and to extend this kind of science cases to other globular clusters in the local group and also extragalactic globular cluster. So this is the end of my talk, but I just wanted to briefly advertise this workshop that is coming up. I'm organizing a calibration workshop next year. A Space Telescope is exactly in the same week, October 14, 18. So since we are talking about surveys and a lot of people were talking about photometric and flux calibration, I bet that a lot of you are interested in this topic. So we have tons of observation coming from all the space telescope. We have LSST coming up soon. We have ELT that will be available hopefully very soon. We have Raman that will be launched in 3 years. We have JWST that is new. So we really need to be careful about performing cross-mission flux calibration and verify that the calibration is homogeneous. So in this way, we can actually match all this data and perform amazing science. Thanks, everyone.